All right. So while we're getting started, just a couple more things to mention. Anybody who couldn't hear, Phys Lab after class, help out with an experiment if you can. For course-related announcements, I'd just like to make sure I put a quiz for this weekend up on Brightspace. Sorry, it didn't open yesterday, but it should have opened this morning. Can people see that? Can anyone see that? No, can't see it. Okay. All right. I'm going to do it right here. Make sure that you can see it so that you have some practice questions for this content we're about to go through. And the other thing is that I uploaded a file that has a bunch of kind of practice study tables in it. So it's got like a blank table and then the answers kind of summarizing some of the major topics that we went through. So I will put up a practice exam for you to practice with over the weekend as well. I need to do a little retooling of that to make sure I know exactly where we get through. So that'll be up for you. And I think actually for Monday, I won't put up a video of more content because our next couple topics I think are worth talking through in person together. So you should also have Monday to study that and go through the practice exam and everything. So that should be good. Okay, so this one is not visible. It is visible? Okay, cool. Thank you. All right, cool. All right. So we're going to finish up thinking about water. Okay. So I want to make sure that we're on the same page about osmolarity, which we were talking about on Wednesday. So let's answer this question for me real quick, because this is going to be important for what's driving the reabsorption that we're thinking about as we're thinking about water reabsorption regulated in the late distal tubule and collecting duct. Let's take a quick look at that. Okay, so osmolarity, when we were talking about the loop of Henle, this was that number where we have an osmolarity gradient going from a lower number up by the cortex to higher as you get towards the renal pelvis. So it's important to know that that high number is high because we have a lot of impermeable solutes in those regions. So when we have a lot of solutes, they act as a pull on the water. So our water is always going to be moving towards those highly osmolar regions, so towards high numbers, because it's moving towards those regions where there's a lot of solute and therefore relatively less water. So think of those numbers as like the bigger the number for the osmolarity, the bigger the pull on the water. Okay, so I'm glad I asked that so that we reviewed that. Okay, so as we're talking about the reabsorption in the later parts of the nephron, so the late parts of the distal tubule, and as we move into the collecting, which is where we have all the nephrons kind of dumping together, we're traveling back through the medulla, right? So we set up the properties of the medulla with the loop of Henle. Here's our late distal tubule, so the end of the nephron. So we're up in the cortex, and then we're diving back down through these regions where we have higher and higher osmolarity. So there's that gradient we see here. So starting at 300, getting bigger and bigger and bigger as we move towards the renal pelvis. Okay. So that's the setup for the surroundings of these areas. Okay. So the way we're able to regulate our water reabsorption with this hormone ADH, all right, is what we're looking at is basically the wall of the nephron here, right? So the part that's neighboring first the cortex and then the medulla. We're going to be changing the properties of that membrane, the properties of that wall, so that we either let water cross or we totally block it. So that's what we're doing with the aquaporins we mentioned at the end of Wednesday, right? We're either allowing water to move into your bloodstream that'll be neighboring the nephron and the collecting duct here, right? So that would be the reabsorption. Okay. Or we block it, right? In that case, no reabsorption can occur. And so we just have our fluid continuing to move out and getting excreted from the body. Okay. So in this picture, in fact, we have an example of what would happen when we don't allow any water to move through the wall of the nephron there. Right, so this is a situation in which we don't have any ADH, don't have any of this antidiuretic hormone. So when we don't have any, we're going to keep our fluid, our filtrate, the same as it moves out of the kidney and becomes urine, right? So we can see as we're moving through, this tubular fluid is at an osmolarity of 100 up here in the distal tubule. As it moves forward, still 100, still 100, still 100, comes out, and your urine is going to have this really low osmolarity, right? So the low osmolarity is going to mean not very much solute, lots of water, right? So low numbers of osmolarity means lots of water, not much solute. Okay. So this is going to have an effect on the volume of urine as well, right? So if you have lots of water in your urine, there's going to be more of it. If you only have a little water, because you're reabsorbing a lot of it, you're going to have relatively less urine output here. Okay. So this is why the permeability of those cells in the distal tubule and the collecting duct with respect to water matters, right? Because sometimes the water is going to be able to leave these tubules and sometimes it's not. Okay. So the way that that reabsorption is happening is because we have this osmolarity gradient, right? So we have these higher and higher numbers, right? So we have a greater and greater pull on water. So when water is able to cross these membranes, so when they are permeable, when we do have ADH present, it's going to be putting aquaporins, specifically aquaporin 2, into the apical membrane of the cell. So that's like the top part, right? Aquaporin, think aqua for water, porin, literally a pore for water to go through. So if we have these pores for water to go through, we have a permeable membrane. So the water is going to leave, going to the bloodstream, and that's going to be concentrating our urine because we're removing water. Now we do have a maximum urine concentration, a maximum osmolarity that you would see in urine. And that number matches our maximum number of the osmolarity here as we approach the renal pelvis. So our osmolarity gradient goes to 1400. So our maximum urine concentration is 1400 as well, because it's that medulla, that osmolarity of the medulla that's pulling water. And if it only has like 1400 power to pull, you can't get above that anymore, right? So you can come to an isoosmotic state as you move through that region, but you can't exceed that because there's nothing to pull more water out of it. Now, different nephrons do have different depths, like how far they go down into the medulla. So it does happen to be the case that you can, I don't know if you're actively changing this during your lifetime, but how much you're going to be able to reabsorb water from any particular nephron is going to make a difference, or what's going to make a difference to that is how deep the loop of Henle went into the medulla, how long it was, because it's that loop of Henle that's setting up this gradient, right? So if you have a little short one, it didn't get that deep, it didn't have that long to set up this really strong gradient, so it wouldn't be as strong. So here's that picture from the previous slide again. So this is our situation where we have No ADH, no antidiuretic hormone. So we're not reabsorbing any water. So we're ending up with all that water staying in your urine. So high volume, but keeping that low osmolarity that it starts with. And here we have a picture of what happens when we do have ADH. Right, so when we do have ADH, water can leave the tubule, can leave the collecting duct. So it does. So you can see that as you go down the length, we're basically matching up osmolarity as we go down, right? So the osmolarity of your urine comes to equilibrium with the osmolarity of that part of the kidney that you're passing through. So water is leaving, 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 leaving until we get to those high osmolar regions as we're moving out of the kidney. So if we do this, we're able to move water across the cells in these regions. Then we're taking water out. So you're going to have that low volume urine. It's going to be super concentrated. It's going to have lots of solutes, not very much water. So it's going to have high osmolarity. Okay. So we know there's kind of a maximum on how osmolar we can get because of how we're setting up the properties of the kidney. And we might remember, and we briefly mentioned when we were talking about pH, that there is a minimum volume of water that you have to excrete. So that's another kind of good reason for not removing all the water that exists in your nephron, because then you just have like chunks of solute, chunks of whatever it is you're trying to excrete, whatever waste products you have. So we need some water volume in there to efficiently remove all the waste that your body is producing and putting into the blood during the day. So that maximum osmolarity matches the kidney, 1400 milliosmoles, and our minimum water loss, so the minimum volume of urine that you should be excreting out per day, here from the kidneys as urine anyway, is going to be 440 milliliters. So these slides are just going to say that same thing, just giving you more bullet points, more kind of words for reinforcement here. So ADH is antidiuretic hormone. What it's doing is it's controlling water reabsorption. So a way to kind of remember what that word does, if you've ever heard that like coffee is a diuretic, right? What people mean is like it makes you pee or something like that, right? A diuretic makes you eliminate higher volumes of water in your urine. So this is anti-diuretic, right? So you would pee less urine excreted because we're stopping this diuretic action. And if you're not expelling that water as urine, it's got to be going somewhere. The somewhere is it's coming back to the blood. At least that's how I remember that ADH is increasing that water reabsorption. Okay. So it's increasing that reabsorption because it's affecting the permeability in the late distal tubule and collecting duct. Specifically, it's helping you insert those aquaporins that you need to be able to get water across the membrane. Okay. And specifically, the aquaporin we're interested in is aquaporin 2. So a reminder of where that was, right? So if we have our cell, our basolateral membrane is this flat one alongside the blood. Apical membrane is the one that's going to be touching the fluid. So we always have aquaporin 3 present here, right? So this is aquaporin 3. So that one we don't have to worry about. It's the aquaporin 2s on the apical membrane that we're going to be varying with our ADH. Okay. So here's how that works. So ADH, we're going to find, is produced by your posterior pituitary, and that's in a bullet point later on. But that is to say, it's going to get into your bloodstream elsewhere in the body. We're producing it from signals essentially in the brain. And so ADH is swimming along in your bloodstream. And as it swims past these regions of your kidney, and we can see it here, we're going to have receptors for it on the cells of the kidney itself, right? So this is going to be a principal cell in the distal tubule or in the collecting duct. Okay. So ADH is present, we can sense it, binds to this receptor, happens to be a G protein coupled receptor, which means that G protein then breaks apart, moves over, and helps catalyze or increase another reaction. So basically what we're going to be doing after we bind ADH to this receptor, so we're going to have a series of steps that eventually lead to the insertion of those aquaporin 2s on the apical membrane. So I don't expect you to know the exact steps in the middle, but they exist. So here's our signaling pathway. We got some stuff going on here, got some stuff going on here. Our total effect, right? So here's that signaling pathway inside. And here we can see that we had a bunch of aquaporin 2s. That's what these blue things are. Right? And they've basically been chilling in the cell, kind of in a vesicle. So they've been waiting to be inserted into the membrane. Okay. So now that we have the ADH present and it's sent us the signal, right? So some of them have been waiting. Maybe we made a couple more. Now we can insert them onto the membrane. So this is kind of like exocytosis, except for we're not expelling them and secreting them totally into the filtrate. We're just binding in the wall of that vesicle, that little bubble. It's bursting like a soap bubble on that apical membrane and then spreading out. So then we have our aquaporin 2s present in the membrane, which means that we have a pathway that water can go through now. So here's that water going through the aquaporin 2. And here's that aquaporin 3 that's always there that lets water out the other side. Okay. So to summarize and kind of link this up to that osmolarity business and our directional words for movement in the kidneys, let's think about how we might summarize what's happening with this presence of ADH. So are we increasing? Do we have high or do we have low water reabsorption happening when we have ADH? And what does that mean for our water excretion? So the water excretion would be that. You know what? I'll just make this a normal mic. Okay. So with that water that we are expelling as urine from the kidneys. So basically we're trying to link up that reabsorption. No, this doesn't work so great. Okay. Less hand movement. Linking up that reabsorption to the amount of urine we see. Okay, great. Yeah, so when we have the ADH, we soak up more water out of those kidneys. And that means that our excretion rate is going to go down. We're going to have low volume urine because that water went somewhere else. Okay. So that ADH itself is regulated, right? We need a signal to tell us, do we want ADH in our plasma or not? Do we want to use that ADH to be turning on these aquaporins or not? So that's coming from the hypothalamus interacting with the posterior pituitary. So the ADH itself comes from the posterior pituitary. So the posterior pituitary is hanging below the hypothalamus from A&P1. Remember, we had like a gazillion hormones from the anterior pituitary, lots and lots of them. Right? We only had 2 coming from the posterior pituitary. So we had oxytocin and ADH. Okay. So those are being released from the posterior pituitary. They happen to be neurohormones because it was the posterior pituitary where we basically had like nerve cells that start in the hypothalamus and then end in the posterior pituitary. And so these cells are going to be reacting to signals coming from some other cells in the hypothalamus surrounding them. So we have osmoreceptors. So osmo for osmolarity in a place called the organum vasculosum of the laminae terminalis, or OVLT. So that's in the hypothalamus, different cell, it's a receptor. Okay. So these cells in the OVLT can sense the blood. So usually in the brain, we have a blood-brain barrier. We don't want stuff to be able to touch the tissue of the brain itself for the most part. But because with these osmoreceptors in this region, we're trying to sense the osmolarity of the blood itself. We're sampling the blood. In this region, we can touch the blood. The blood-brain barrier isn't really in effect. Okay. So when we get a signal from these osmoreceptors telling us that the blood is getting kind of highly osmolar, right? So it's getting a lot of solute buildup, which means not very much water. That's what's going to signal this production of ADH, trying to get more water into the bloodstream to balance that out. Another thing that ADH may be reacting to are baroreceptors. So our baroreceptors, we might remember, they're sensing our blood volume and our blood pressure, right? So like the amount of blood in the veins and your mean arterial pressure, basically your blood pressure. Okay. So the reason for that is going to be that if you have a drop in blood volume, right, most of the volume, therefore most of the pressure in the blood is coming from that presence of water, right? Your blood is mostly water. So if we have low blood pressure, low blood volume, we'd have low baroreceptor activity, which is going to be a signal. We want more water in our blood. We want higher blood volume. So we're going to start reabsorbing a bunch more water by turning on this ADH secretion. Right? So we're turning on the signal to the kidneys that we want to reabsorb more water. So we can see this in flowchart form too. Right? So this one is just talking about that osmolarity and its regulation of ADH. So if we have highly osmolar extracellular fluid next to a blood vessel, next to these osmolarity receptors in the hypothalamus, we're detecting all those solutes and we're responding. So we're sending signals, signaling to the posterior pituitary. It's time to make a bunch of ADH. And then that ADH is going to go tell the late distal tubule and the collecting duct to insert all those extra aquaporin 2s into the apical membrane so that we can reabsorb a bunch of water. Right? We reabsorbed it, so we're not going to excrete it out. So that water excretion goes down, meaning that we're going to conserve any water you're drinking or producing metabolically. This should hopefully bring that osmolarity in the blood and therefore in the extracellular fluid back to our normal expected range. Right? So once we have more water in the blood, it's not going to be as osmolar. And we got a similar thing, similar flowchart going on with our mean arterial pressure and our blood volume. So if either of those drop, and I can't think of a situation in which one would drop, but the other would go a different direction. Okay, so basically, if both of them drop, blood pressure, blood volume, we have receptors, pressure receptors that are sensing that. Okay. They're affecting the hypothalamus. Hypothalamus says we don't have enough blood pressure, we don't have enough blood volume, so we make ADH, we reabsorb water, so we're not excreting it out, which means we're keeping that water in our blood and we're hopefully bringing that blood pressure and that blood volume back up. Okay. So recalling and kind of summarizing. So if we don't have this signal going, right, we've effectively fixed our problem, or we just didn't have a problem in the first place. So our posterior pituitary isn't making this ADH. What's the urine going to look like? Okay, let's think about that volume, right? So if we're not making ADH, that means we don't have ADH, we don't have this hormone in the plasma, which means it's not signaling to those cells in the kidney. When it's not signaling to the cells in the kidney, water is going to be blocked from traveling across those principal cells because we don't have enough aquaporin 2s. So water isn't getting reabsorbed, so it's got to stay in those tubes in the kidney, which eventually come out of your body as urine. So that's why we have this high volume urine in this situation, because we didn't reabsorb the water. It just stayed in the kidney, so it went out the kidney. This is going to be low osmolarity because when we have a lot of water and not as much solute dissolved in it, that's what low osmolarity means. This is going to work in exactly the opposite way if we wrote the opposite question. So if we are secreting ADH, it would be low volume, high osmolarity. So that other one's the opposite. Okay. So we got one final piece here to tack on to our understanding of what's going on with our water balance and the kidney. And we're going to think a little bit more about our glomerular filtration rate here, our GFR. So remember, our glomerular filtration rate is essentially telling us how much water are we filtering into the nephron, into the tubules in the kidney per minute. Okay. So we're going to see a link between glomerular filtration rate and blood pressure. And we're going to see a link between that glomerular filtration rate and what is actually coming out of your body at the other end of the kidney. Okay. So we talked about regulation in this kind of normal range of our glomerular filtration rate, right? We had this big plateau, and we could do that because we had 3 different mechanisms for trying to keep our glomerular filtration rate in place, right? So we had our, for example, the smooth muscle in the afferent arteriole, if it stretches out when you have high blood pressure, bounces back. We have other ways of regulating our glomerular filtration rate, which we talked about before, but that's one of the main ones. So that's the auto-regulation piece. Usually we're doing this local regulation of glomerular filtration rate, but we're going to see effects on the glomerular filtration rate, particularly if your mean arterial pressure drops too low. So particularly here is when we're going to see effects of our mean arterial pressure on that glomerular filtration rate, and therefore also on what is coming out of your kidney. So when you drop to these low levels of blood pressure, right? So low MAP, this means that you have not very much pressure coming into any of your blood vessels, including into the afferent arteriole and into the glomerular capillaries. Now, the hydrostatic pressure in those capillaries was one of our big drivers of filtration, right? So that was one of our Starlink's forces, right? So if we have this low blood pressure and therefore this low pressure in the capillaries, This means we're not going to have as much water moving into the nephron. So we're going to see a decrease in the water that gets filtered in the kidneys, that gets filtered into the nephron in the first place at the kind of start of our system. If we have this decrease in water being filtered, this means we just don't have as much water in the nephrons in the first place. So when we're at these really low levels, beyond our auto-regulation range, this means that since there wasn't that much water going into the kidney, there's not that much water that can go out of the kidney. So we're going to see a decrease in the water that gets excreted as urine when we're at these really low levels. And we would see kind of the opposite effect if our blood pressure got really, really, really high. All right. So this low range is low, but not crazy low. This high range is kind of crazy high for a mean arterial pressure. But if it were to happen, this means that we have basically a fire hose of pressure coming into the afferent arteriole. So we're going to have tons of hydrostatic pressure in the glomerular capillaries. So we're going to have a really high glomerular filtration rate as a result. So this means we're going to have tons and tons of water coming into our nephrons, coming into the kidney. And so that tons and tons of water is going to kind of blast through, and we'll see an increase in the water that gets excreted as urine here. Now, this in particular, you're only really going to see in pathological circumstances. We should be able to keep ourselves at least in the top part of this range. Okay. So we're going to talk about sodium and potassium next. We'll see how far we get. Now, the way that sodium balance and potassium balance work is really similar, really analogous to what we've just been talking about with water. We're just going to have different hormones that we're paying attention to and some slightly different mechanisms if we look at the details of what's happening in the cells. Okay. So like with our blood volume, our hypervolemia, hypovolemia, we have a word for sodium and one later on for potassium. So hypernatremia, look for this Na in the middle. Na plus is sodium. Okay, so lots of sodium is hypernatremia. Not enough sodium, hyponatremia. Now, sodium is really important. Regulating it is really important because this is our main solute in the extracellular fluid. So this is really important for establishing our baseline osmotic pressure. That was one of those main solutes, our main ions that we were pumping, like in the ascending loop of Henle. We know that sodium is really critical to how your neurons work. It's really critical to how your muscles work. We have sodium concentrations really driving a lot of processes in our body. So the excitable cells, our nerves, muscles, but sodium is really important everywhere. So we want to keep our sodium in balance. We want to keep our sodium levels at kind of regulated, consistent state, so your cells can vary those sodium levels in order to have function. Okay. So when we think about what's happening with sodium, so this is one of the things that is freely filtered along with almost everything else, except for proteins in the plasma. So that means that the sodium that's coming through your glomerular capillaries is just moving straight into the nephron. There's nothing blocking it there at that filtration step. As it travels through the proximal tubule, we saw that lots of sodium is getting reabsorbed immediately. And remember that sodium reabsorption and reabsorption in general in the proximal tubule is unregulated. So that's not where our hormone is going to come into play. It's in the distal tubule and the collecting duct that we're going to see regulation of sodium reabsorption using a hormone called aldosterone. We don't have to worry about hormone or our sodium secretion. So we're just going to be thinking about reabsorption into the bloodstream and a little bit about excretion out of the body in the urine. So about 70% of your sodium is usually reabsorbed into the blood, kind of combined between these three locations. So 30% of it is kind of staying in the urine. Okay. So the main hormone that's going to be involved here is aldosterone. There's another hormone called atrial natriuretic peptide, A&P, that comes from your heart. I don't think we'll get to A&P today, but that's fine because aldosterone is the main important one. So this is going to be happening in the principal cells, in the distal tubule and the collecting duct. It's going to be happening actively, aka using energy, using ATP. And it's going to be happening regulated. The regulation part is the hormone. The sodium potassium pumps on the basolateral membrane are going to be driving our reabsorption like they are in a lot of places. So a reminder what was happening in the proximal tubule. We mentioned it when we were focusing on the proximal tubule as a region, but our takeaway with respect to sodium was our sodium potassium pump creates a low sodium region here inside our epithelial cells in the proximal tubule, which means we've got high sodium here in our peritubular fluid and relatively high sodium that's getting freely filtered into our tubular fluid. So this creates a gradient that allows sodium to move into the cell. And we know that sodium and its movement is often paired to other things. It's often dragging stuff with it, like glucose, or potentially, by the time it gets here, attracting some water, attracting some chloride ions, right? But it moves passively down its gradient into the cell, and then we pump it into the peritubular fluid. This is going to be high concentration, so then we can kind of move passively into the plasma, which again is going to be relatively kind of low. Okay. So that required some energy, but there was no hormone involved. It was unregulated. In the distal tubule, we have some reabsorption that looks similar to that, right? So we have a sodium potassium pump. This means we're creating low sodium in the cell. We can see that we use that sodium to move some things into our distal tubule cells. We also have some channels that are just for sodium here. So here sodium really is moving through passive facilitated diffusion here down its gradient, and then getting picked up by this sodium potassium pump being pumped out. The regulation piece is going to come into play in the principal cells in that same location as we just saw in the previous slide. So a different type of cell, but still in the distal tubule and in the collecting duct. So these cells are going to be reacting to aldosterone just like they were kind of reacting to ADH. Okay. So what happens is we have a steroid hormone that's coming from your adrenal glands, specifically from the cortex part, so the outside part. So we saw that the adrenal gland, the inner part, was kind of linked to our sympathetic nervous system, was producing lots of epinephrine, a little bit of norepinephrine. Adrenal cortex, one of the things it's doing now for us is making aldosterone. Okay. So this hormone, just like ADH, has a receptor. A difference that we might notice is that the receptor is in a different location. So aldosterone is a steroid hormone. Hormones generally are either steroids or proteins. But this is a steroid hormone, which means it's a lipid, nonpolar, can cross into the cell. It can actually get in there without traveling through a channel, without having anything facilitated. So we put our receptors on the inside of the cell, just kind of floating in the cytoplasm. When we trigger those receptors, though, we're going to have an effect on what's going on in the apical membrane, so the part that's touching your tubular fluid. We are going to open and to actually actively make a bunch of sodium channels. We also, with the same hormone, aldosterone, when we talk about potassium, are doing the same thing for potassium, but the potassium gradient goes the opposite way. So when we have potassium travel, instead of moving towards the blood, it's going to be moving away from the blood. So we can see here, aldosterone opening up these sodium channels. It would also be increasing how many sodium potassium pumps we have here on the basolateral membrane. So it's making this gradient really extra noticeable, right? So we're making the sodium especially low here in the cell. I don't know, maybe plus isn't a great thing to draw for low, but super low because we're pumping that sodium out fast, meaning we're able to pull lots of sodium into the cell using that gradient, and then we're able to pump lots of sodium into the blood because that's the point of the aldosterone here, is to get more sodium reabsorption into the blood. Okay. Now, where the aldosterone comes from and how it's being produced, how it's being regulated is, I think, a little bit more complicated potentially than the ADH. Involves more organs anyway. Okay, so our key players that we want to notice here is we have cells in the kidney, those juxtaglomerular cells, so juxta next to the glomerulus. So they were in this region where we have kind of the end of the nephron going into the distal tubule, and that's coming into contact with our arterioles that were going into the glomerular capillaries. So we mentioned this as part of our regulation of glomerular filtration rate. This is also playing a role in this sodium reabsorption. Okay, so those are our juxtaglomerular cells. Specifically, there are going to be some granular cells, is the name of the specific cells in this region that are going to be responsible for things. Before we get through our kind of workflow, I also want to point out the liver is doing stuff here too. So the liver is making another protein that it puts into the bloodstream, just like all those clotting factors we saw. We have basically an inactive protein, so that inogen ending is kind of a clue to us. This is called angiotensinogen that the liver is making. We know that aldosterone, which is the thing that we're trying to regulate, is coming from the adrenal cortex. Okay? So those are kind of our three outside the bloodstream features that are going to have to work together to get that aldosterone coming to the kidney. We're also going to see that even the walls of your bloodstream itself, so the capillary endothelial cells, the lining of the capillaries, have a structure that's going to play a role. So these are some enzymes that are embedded in the wall, basically. So these ACE enzymes are also part of this kind of workflow. Okay. So what's happening here is basically that we're going to want to take that angiotensinogen that's in your blood. We're going to want to convert it to angiotensin 1. So we dropped this inogen ending because inogen kind of tells you it's inactive. So we're starting to activate it to angiotensinogen 1, then to angiotensinogen 2, and then it's going to turn into aldosterone. So this is what we're trying to do. Okay. And we have these ways that we basically catalyze these reactions, right? So this conversion of angiotensinogen to angiotensin 1, renin is doing that. Conversion of angiotensinogen 1 to angiotensinogen 2. Those ACE enzymes on the capillary wall are doing that. Then that angiotensinogen 2 is coming into the adrenal cortex, and the adrenal cortex is stimulating the production of the aldosterone. And this is just that picture in big form. All right, so angiotensinogen should always be in your bloodstream from the liver. When the juxtaglomerular cells produce renin, and those granular cells in this juxtaglomerular region produce renin, that's our signal to activate that angiotensinogen into angiotensin 1. Once we have angiotensin 1, we don't have to wait for another signal. We've got our ACE enzymes in the capillary endothelial cells that are going to convert to angiotensin 2, going to the adrenal glands, and their cortex produces the aldosterone. Okay. Oh, I didn't mention the title of that previous slide. Okay, so there's a name for this process. Can I move backwards? Yeah, okay. So the name for this process is the RAS or renin angiotensin aldosterone system. Okay. So it's naming basically what we're doing, right? We're using renin to produce those two types of angiotensin to trigger our aldosterone secretion. Okay. So here you can see some flow charts of how this relates to our mean arterial pressures. For now, we'll go through the details of this later because this is a little complicated and we got like 1 minute. But you will notice this in your study tables, just so I'll mention. When we have this angiotensin present, secrete aldosterone, increase the amount of sodium that's being reabsorbed and therefore moving into your blood. We now have high osmolarity in the bloodstream. So we have a domino effect where this goes to the posterior pituitary, triggers ADH. So we trigger water reabsorption, which means we reabsorb more water. And so our blood pressure goes back up. So in that table, you'll see that these hormones do eventually kind of play with our blood pressure, with our mean arterial pressure, because of these kind of interconnections. Okay. So we will leave it there. So do know how this RAS system works. Do know how aldosterone is affecting these distal tubule and collecting duct cells. You can leave the blood pressure stuff, or we'll spill that over into the next exam.